Hi, Malcolm. Can you hear me okay?
Yeah, I can hear you, Harold, and you don't sound as fuzzy as yesterday.
I had to use the browser version this time. I could not launch the app.
Is it updating or something?
Yeah. Shouldn't be. I'm supposed to be told when these things happen.
Hi, everybody.
Hello.
Hello.
Hello, good morning for those of you who's morning, and hello.
Morning, Petra.
Oh, it's morning.
Sorry, West
For
Coast.
some of you, it's very early. Yeah.
Yes, it is.
3 hours of sleep.
Well, I just had a nap to make it until 8.
Yeah, right. When we had the Go meeting, I was in Germany, so I know how that feels. So, but it's nice, Paul. You have still already good light, so that's good.
Yeah, the sun's up. It's not too bad. We're pretty far south here in California, so
So are all the fires done with?
for the time being,
I noticed we don't see anything about them on the news anymore.
yeah, no, the air quality is good again, at least down
Okay.
here. Yeah, it has been for probably a week or so.
That's good.
Yeah, it's really nice. It's nice to be able to go outside again and feel okay afterward.
Sun here, but it's a bit on the cool side now. We had
Hmm.
actually over almost 10 days of, they call it now, oh, it was nice to have the November summer. We
Yeah.
had really amazing. And when the election was called on Saturday a week ago, it was still so warm. We were at the farmer's market at that time, and the market exploded in joy and dance and everything. It was really fantastic, actually, in town.
That's great.
We've got a bunch of cold Canadian air coming down on us any second now.
Hmm.
Yeah, and we
Okay.
get some of that.
Then I won't tell you it's going to be 80 degrees here tonight.
Yeah.
I'm not going to say a word about it.
I figured for this virtual jamboree, just decide where you want to be, right? And that's what you can do.
Right.
Okay, so it's 4 after the hour, so let's go ahead and get started. I imagine other people will join us at some point during today's session. So first off, thank you everybody for coming yesterday, for taking part in the discussions, asking great questions. Starting today, we really want to get into more of the hands-on sessions of the GO-CAM Jamboree. So this is an opportunity for everybody to really try their hands at Noctua, try to create some basic activity units and some basic linking between them. So what I thought I would do this morning is, since I know we still have people who are relatively new to this whole process, I thought I would just again go through the goals of the workshop and then a really hopefully straightforward example of how to create activity units using the form, and then how to link them up in the graph editor and put evidence on that. Again, just as really sort of a refresher so that everybody can follow that process, since that's basically what we want people to do for the workshop this week. Before I get started on that, does anybody have any general questions that they want to bring up? And certainly if you do during the course of this, I also said yesterday, I put a section at the bottom of the agenda for questions, and Colin already put one there, which is great. Feel free to go ahead and do that, and we'll make sure that we address them.
Question, Kimberly. Are the steps you're about to go through written out someplace on some kind of a cheat sheet document?
Yeah, so I have them actually in a video feeder. So what I'll do, and the video is in the Google Drive, so I'll link that actually to the agenda. Eventually we want to move all of these onto a Go YouTube channel, but I'll link the video
Oh, great.
from the agenda.
Great.
Yep. Yeah, and the script. I'll put the script there because I wrote it all out as a
And you don't check users, so we can go there and refresh our memory without embarrassing ourselves. Okay.
No embarrassment. No one should feel embarrassed about that, right? This is what we really want this week to be. You know, we realize people don't do this all day long, every day. And so this is an opportunity just to become more familiar with it. So please do. Please ask questions. Please check those videos as much as you need to. Okay, so let me share my screen. And I'm just going to turn off my video. Do this. Okay. So here we go. And then, so I just want to, I'm going to log myself out. So I start from the very beginning and things. Okay. So again, what I want to go through this morning is using Noctua. And again, it's really just the basics about how to use the tool and get started. So just a reminder also of everyone of what the goals of this workshop are. Like I said, number one, we want people to be more comfortable using Noctua, the landing page, as well as the form and the graph editor. And as Paul presented yesterday, you know, we want people to be confident in understanding the general principles that underlie GO-CAM modeling. We want people to be aware that there are different causal relations that you can use, but we think not so many. And we want people to come away with a good understanding of how they're used. And if by the end of the week you've got the beginnings or a full-on process-centric model, that would be great. But mostly we want people to be comfortable creating the building blocks for those models. And then lastly, we welcome feedback, you know, on the workflow, the tool, and especially as Paul said, you know, give us an indication of the time that you spent in creating a model, so that we have a better idea of what we can project that we can do for the next grant renewal. I was taking a lot of notes yesterday on the comments and the feedback that we got, and I'll try to do the same today. Okay, so I'm going to illustrate again how to create really basic activity units in the Noctua form, and then how to create a causal relation between them in the graph editor. So here's a paper, a relatively recent C. elegans paper that I'm just going to use to illustrate this, right? So the two activity units that I'm going to create are one for a gene called UPL4, and the second one for a gene called DVE1. And both of these are involved in the mitochondrial unfolded protein response, for which we know quite a few things, but there are also quite a lot of the details of this that still need to be worked out. So this is just going to really be the beginnings, or the building blocks of this model. So to make it really simple, remember, this is essentially what I'm going to do. The process is going to be the mitochondrial UPR. And basically what I'm going to say is that there's an activity enabled by this UPL4 gene. This happens in the cytosol, and that has a positive regulatory effect on the activity of DVE1, which actually is a transcription factor. So it actually has to go from the cytosol into the nucleus to enable its activity. So I'm just going to create the two activities, where they happen, what process they're involved in, and how they relate to one another. Okay. All right, so let me go back and I'll start at the very beginning for Noctua. And so as we said yesterday, when you first go to Noctua, this is where you'll go, which is the Noctua, what we call the Noctua landing page. All right. So I'm going to create a new model with this example. So the very first thing I have to do, again, if you're not already, is just log in. So I'll click on the green button, and then I'll sign in with GitHub, which is what we use for authentication. When I see that it knows it's me, I'll just go to return, and then I'll be back to the landing page. And now you'll notice I have the two options here that I can start creating a model in the form or the graph editor. So in this example, I want to create a new model in the form. So I'll click here. And that will open up a new tab of the Noctua form editor. Okay, so this is what basically a blank slate looks like for creating a new model with the form. So I want to create 2 activity units. So to do that, go up here and click on Create Activity. And that will open up this interface that has the pre-populated fields and relations between the fields for an activity unit. So let me start with the upstream gene in this case, which is ULP4. So this will autocomplete, and as we talked about yesterday, I annotate to gene ID, so I will pick this one from the list. Okay. So the molecular function in this paper, they show that it's has sumo isopeptidase, sumo-specific isopeptidase activity. So we talked about yesterday that you really have 2 options for adding your annotations. And we encourage people to, as much as possible, use the annotations that already exist, right? So in this case, if you want to look to see what's there, just go over to this circle and 3 dots for more actions. And we have a search annotations option, and that will give you a list over here on the left of all the function annotations that currently exist. So the interesting thing here is that right now, the more specific molecular function that I want to use for this model, we currently don't have, right? So that's fine. What I can do then is I can just add a new function annotation. So this is essentially just adding, just like we would for standard Go annotation, I'm going to make a function annotation. So in the function box, sumo-specific, and this is the term that I want, sumo-specific isopeptidase activity. And this is just from reading through the examples in the paper, okay? So for evidence, there's a direct assay in the paper, so I'm just going to type IDA, and then I can pick the correct Go evidence code. I want to add the reference. So as we said yesterday, there's a nice little check in the form. This is a PMID. I'm just going to type in my PMID. And it will go out and double check that this is the reference and this is the paper I want. So then click on the checkbox and we'll add it. Now I know also from what I just read in the paper and what I showed you is that there's an input to this. And so again, just like our normal conventional annotations, I can add an extension to that. And I want to add a has input. And the has input for this is DVE1. And again, from the list, I'm going to pick the C. elegans gene, right? And the evidence is from the same assay, okay? So again, what I can do is I can just clone the evidence that I've already used. So I'll select that and add it. So I don't have to add the same thing manually each time. And it will show you I've added my function and here's the extension for has input. So same thing for biological process. We can look to see what's there. And I don't have a mitochondrial unfolded protein annotation yet, so I can't add from an existing.
Kimberly, may I interrupt with a question for
yeah.
a second?
Mm-hmm.
So you've picked direct assay evidence used in manual annotation by typing IDA,
Right.
but when reading the paper, you probably saw that it was an enzyme assay, right?
Mm-hmm.
Is there any reason not to choose the more specific eco term enzymatic assay?
Mm-hmm. Yeah. So yeah, sorry.
Assay used in experiment, what's it called? Enzymatic activity
Yeah.
assay used in blah, blah, blah.
Mm-hmm. Yeah. So, I mean, full eco is available to curators in Noctua. We're not using it yet at Wormbase. And I think that, you know, one of the issues is that the Go consortium on a whole, we still produce a GAF, right, for people, and GAF uses the 3-letter codes. So if
There's a mapping file.
yeah, no. So if, right, if you know, you want to do that and you know that it can map up the evidence, the more specific evidence code that you chose maps up to a 3-letter code, you're certainly free to do that, but we're not requiring that curators do it at this point. Does that
Okay,
answer
but
your
it's not
question?
just the limited exact match eco that's available through
That's right.
Noctua.
That's right. Full
Mhm.
eco is available in Noctua. That's right.
Okay, thank you.
Yep. So it's really up to the individual groups to decide what they want to do. And just, yeah, I would just make sure that whatever you choose can still map up to a 3-letter code.
Yeah, and while we're on the topic of evidence, you know, as you start to build your, you know, a first draft of a model, and even something that, you know, is that you might want to release at least at first, it's fine to use, you know, let's say you're working from a review, it's fine to use TAS evidence codes. And so just to remind everybody that we, you know, we do still have all those evidence codes available in Go, and that, you know, if you haven't had time to track down actual experimental evidence, you know, TAS, traceable author statement evidence, is fine to use. So just wanted to
Yeah.
throw that out there.
Okay. And so just to continue on, this is actually, I'll add the IMP, same paper. In this case, I'll just cut and paste. And then for component, remember that for the activity unit, when we add a cellular component, the statement that you're making is that this is where the molecular function occurs. And so for this, I'll just say cytosol. And as Paul said, we can use PAS. So I'm going to do that for this paper. Okay. And so then you notice that I filled everything in as we want it. And the save button is now blue, which means I've got the information I need. One thing we'll show is that if you want, you can click on this nice preview button. It'll give you a really, you know, quick summary from a graphical perspective of the statement that you just made, which is that there's a function enabled by ULP4 as part of this process. That function occurs in the cytosol and the function has the input. Okay. So now I'm going to just go ahead and save this. And like we said yesterday, once you've saved in the form, you'll start this table view, which will keep track of each of the activity units that you've used. And if you want to, at a later point, edit anything about them, you can do it here in the table as well as in the graph editor. Okay. All right, so now I want to add another activity unit in the form for the downstream gene. So in this case, again, it's DVE1. And I will scroll down and pick the gene. Okay, so again, let's go look and see what exists for this. So in this case, I see that there is actually an existing annotation for DNA binding transcription factor activity, POL2 specific. So I'm going to go ahead and use that. And in this case, it actually comes from a paint annotation, which is totally fine to use. And so then we'll go ahead and populate that. And I'm going to do the same thing for process. We have some that exist, 2 annotations that exist already. I could certainly also add from the paper that I'm curating here. So let's see, I've got a mutant phenotype from that paper. I can add that too. Whoops. Let me try that again. I think that got
You
rid
have to
of,
add
yeah,
evidence first. Yeah.
yeah, sorry. So yeah, let me just do that again. So I'm going to take what exists. There we go. And then I'm going to add evidence, and now we'll clone it. There we go. Mutant phenotype. Whoops. Nope, still doing that. Looks like there's a little bug there, but I'll ask Jermaine about that. We can fix that. And then component. So for component in this case, again, the statement that we want to make is that this is where the activity for this gene product occurs, right? So in this case, it would be the nucleus. Again, I'll search annotations, and we have a whole bunch of evidence for that. You can add as much evidence as you want. I'll go ahead and I'll select them all. Okay. And so again, now the save button is blue, and I can go ahead and save this next activity unit. And then I'll close this, and I have both of these now in my form. Okay. So the next thing I want to do for this model is I want to actually just link these two activities with a simple causal relation, and that's going to be positively regulates. So we can do that in the graph editor. So to go to the graph editor, I'll click on the icon for the graph editor. Like we said yesterday, the two tools talk to each other. So I'm just going to move upstream and downstream here to be left and right. Okay, and all I want to do now is I want to link these two activities. So to link two activities in the graph editor, click on the blue circle in the starting activity, and then drag the box over to the next activity that regulates until it's highlighted, and then release, and you'll get the pop-up window with all the relations. So I'm going to pick positively regulates. Okay. And then remember that every statement, excuse me, every statement that we make, we want to be able to put evidence on that. So for the relations, in order to put evidence on it, click in the box, the empty box here, and you'll get the pop-up window in the graph to add the evidence. And so in this case, I'll just enter this manually, but again, you can clone if you want. So, excuse me, I'll do IDA, and then I'll enter my reference. When that's done, I can just click add, and I'll now have a piece of evidence on that relation statement as well. So, and like we said, whenever you're working in the graph editor and you make some changes, go over to model and to save. And so there it is. All I've done is I've created two activity units, and then I've just linked them up with a single causal relation using the graph editor. So are there any questions about that workflow or how things work there?
Hi, Kimberly. So on the nucleus evidence, you put
Mhm.
quite a few bits of evidence on there.
Mhm.
And some of those were they electronic annotations as well?
Mhm.
So what I'm a bit confused about is, say, like Interpro doesn't update and that particular association disappears. How does that feed into the Noctua ecosystem?
Yeah.
So we do a lot of like asking Interpro to remove associations and things like that. So hoping that it will feed forward into our annotations. So I'm just wondering specifically.
Yep, exactly. Yeah, so basically what Helen's asking is that, you know, with electronic annotations, right, there's some churn on that. Sometimes we ask to update the mappings and things will change. So what we're planning to do in the Noctua ecosystem is just the same thing that other groups do, which is that when there's a change, we'll have, you know, programmatically go in and we can update any of the annotations that exist as part of GO-CAM models in Noctua.
Sorry, I missed that bit when you put the evidences on. So can you go to that section, just where you select the one that Helen's talking about, where the multiple, so if you've got, presumably the IEA wasn't the only evidence.
No, no.
Is it worth collecting the IEA evidence for these when you've got experimental? Does it really add any value? And would that save work to just, I mean, because I mean, as you know, in POM-based, we don't even display evidence. If we've got experimental evidence, we're not interested in the other types. So, and that means, yeah, it just seems, because a lot of these as well, they're going to be, when people display them, if you've got all this evidence to show, it's a lot of stuff to deal with on your gene pages and wherever you display it. I just wondered if you thought it was valuable to transfer the IEAs on a Noctua model if you've got data from a better source.
Yeah. I mean, so basically what I just wanted to show there is that you can add multiple pieces of evidence onto an assertion if you want, right? And I know that some groups may want to, some groups still do now include, right, both IEAs and experimental on their gene pages. And I think it's up to the individual groups to decide what it is they want to do. If you don't want to add the IE evidence, you certainly don't have to, right? You can choose what.
I mean, one would assume anyway that if the IEA is there with the experimental, that it's not really going to churn that much, because most of the IEAs that are in the churn are the ones that are outliers anyway, and there aren't many of them. And that's the same with the IBAs, I guess. They're either going to be all wrong, or it's going to be unlikely that you've got an IEA that's wrong and the other ones are correct. I was just thinking if it would save work, but it probably won't, because they'll be either all or nothing. They'll either all be wrong or all be right. Yeah.
Yeah. No, I mean, really, it's up to, you know, the individual groups.
So would you use IEA as the evidence if there was no other evidence code, or would you look for something that was experimental?
Yeah, so that's a good question, and, you know, speaks to workflow. So again, I think that's, you know, what people feel they have time to do, right? I love going out and looking for the experimental evidence, right? If I know something is missing. But I think at the same time, you know, if there's an IEA annotation that seems correct and specific enough, and I, you know, can't relatively quickly find the experimental, I think it's totally okay to use that IEA. And know that down the road, right, hopefully as experimental evidence comes along, we can add that as well.
Okay, yeah.
Can I just ask, if you want to actually get the sort of exported annotations, do the evidences need to be consistent? If you have like mixed evidences, will those all result in meaningful annotations?
So I'm not quite sure what you're getting at. I mean, hopefully, right, curators would only put the evidence on that they think would lead right to a meaningful and correct annotation.
But just when you were adding them, I thought you added several different types of evidence for different elements. But don't you need to have like a consistent type of evidence for you to actually get a consistent sort of annotation chain?
So are you asking about we might get for extensions? I mean, certainly, right, we know that, right, just the way, right, research goes, that we may not have the same kind of evidence, right, for all the statements that we make, right? And that's okay, right? Again, we'll use the best evidence that we've got, and we can put multiple pieces on if we want. With respect to annotation extensions, and we kind of talked about this a little bit yesterday, you know, it still follows the same principle that our conventional annotations follow, which is that if we're going to add the extension in the GPAD output, it has to have the same evidence as the annotation, you know, to the Go term, right? Since that's the extension really, you know, modifies the Go term. So does that answer your question, Malcolm, or is there something else?
I think so. I mean, I guess like the distinction between what is in like the sort of annoton thing that you're building up here and what is an extension is maybe not 100% clear to me.
Okay.
I mean, you have these three different things. You have your molecular function, your biological process, you have all these things. Or like your sumo-specific isopeptidase activity has the occurs in, the enabled by, and I believe you had different types of evidence in some of those cases. And I'm just asking,
That's right.
which of those becomes an actual annotation, or do they all become an actual annotation?
So the script for the GPAD output, right, will try to create as many standard conventional Go annotations from our GO-CAM models as possible, right? And it does that, and we can talk a little bit about this maybe later in the week about the details. Basically does this by, following rules for how we link Go terms together with the relations in a GO-CAM, right? So just to use this as an example, for GPAD output, I would want an annotation that UPL4 enables the sumo-specific isopeptidase activity. That UPL4, in this case, is active in the cytosol, and that UPL4 is involved in the mitochondrial unfolded protein response. So at the very least, we would get those annotations out in the GPAD. And again, if we're going to also get extensions, the evidence would have to be the same for the term, and the contextual information we added is the extension to get it out. And that just follows our current annotation practice with respect to annotation extensions.
Is there a way to visualize what you've just done as GPAD? Like, can you see what the predicted output would be?
Yep. So there are kind of two options you can do for that. So under this workbenches menu, there's something called the Annotation Preview. This isn't full on GPAD, but it will show you, give you an idea of what annotations you would get out in the GPAD, right? So we need to do some work on updating the rules for the GPAD output to conform with our new specifications for GPAD 2.0. But this is essentially what you can look at to see if what you're getting out, is consistent with your model. It's consistent with what, you wanted to say for this. So you should have, our function, process, and component. The component relation will be updated in accordance with GPAD 2.0. And just notice that the relations, right, in this GPAD output, they're different, but they're different because these are gene product to term relations, right? And so the GO-CAM model for GPAD output gets translated so that the relations that we use in the model get translated into the gene product to term relations for our standard annotation files. If you want to look at the full-on GPAD, go over to Model, and you can actually export the GPAD. And if you click on that, you'll get another tab in your browser that will actually show you what the GPAD will look like.
Kimberly, there's no, I think the chat function is disabled. I was going to put
Oh, okay. In the Zoom?
in the Zoom, just because
Oh,
if whoever's running it could open it, I think that would be good. I have a quick question.
okay.
First, back on the previous screen, when you went to the workbenches menu and you chose your annotation preview, I know this is very trivial, but could you put something like that? It says like GPAD in brackets after that too, so people knew that that's where you can double check what the annotation preview format's going to be. There it says, like you have it on the other export models as GPAD. I don't know, you've got a bit of real estate. You may or may not want to do that, but that would have helped me figure out what the annotation preview was. But my other question is, when you're adding all the evidence, can you remind me what you do? And if the evidence is from mouse and we're working on fish or frog, how do we put the evidence of molecular function through the orthology? How do we do that? Like if we want going to import evidences from somewhere else? Is it going to give us the molecular function of these genes in different species when we go and look for previous things, or is it going to be limited to the species we're working on?
Okay, so let's try to illustrate that in the form. So let's say, do you have a gene in mind you want to try with Xenopus, or?
Yeah, I'd have to quickly.
Or yeah, okay, we can certainly illustrate this. So let's say,
and see if does it show with the evidence from other species, for instance.
yeah. So yeah. Right now, so if I type in a gene product here, let's just type in another one, another ULP, right? If I wanted to make a function annotation, right, based on a function that had been demonstrated in another organism, say mouse, yeast, and we have to do this a fair amount in C. elegans. If I go to the search annotations right now, all it's going to show me is, what exists for that C. elegans gene, right? And so if I want to make the function annotation based on what's been demonstrated in another organism, again, it's just like we do it for conventional annotations. Let's just say, just for illustrative purposes, that. And so then for evidence, you would want to use one of the sequence-based evidence codes, inferred from sequence similarity or inferred from sequence orthology. Right? So let's do, let's do an ISO, inferred from sequence orthology. I'll just put in the same reference just to illustrate. Okay. And then, so then what you have to do is you have to say what you're basing this statement on with respect to orthology. So in the with from field, what you need to do in this case is actually populate with the identifier for the gene from the other species that you're basing this statement on. So let's just say we're going to use a uniprot, maybe, and I would enter that there. Okay. So I think it would be an interesting idea, Chris, if a paint annotation didn't already exist, right? And I often use the paint annotations, right? Because they're based on, the phylogenetic relationships, and they filled in a lot of gaps. But if there wasn't a paint annotation that already existed, I actually think it might be an interesting idea from the form to be able to maybe hop out and see, what other annotations exist, say for orthologs and other species. There'd be some tooling behind the scenes we'd have to do for that, but that might be a helpful thing. But for now, this would be the workflow.
Okay, thank you.
And Chris, one thing to bear in mind for us specifically is you may have to look at trop or Levis or the S or the L to actually get the annotations you want.
Right.
They're not necessarily all to one thing.
Right, because we have three genomes to deal with. All right, thank you.
Sure. Are there any other questions?
Yes. Hi, Kimberly. I'm trying to add some toxin and to make some annotations to a toxin protein, right?
Okay.
And it is not recognized at all. The thing is, it's not a gene, so it's like identifier. So I'm trying to put uniprot, KB, the dot, and the accession number, okay. But it's not recognized at all.
Okay. So what's the species, Penelope?
It is from a snake, I think.
Okay.
Yes.
All right. So probably what's happening there is that we don't have all of the uniprot accessions available in Noctua.
Yeah, I think what we worked out, right, wasn't it that that it would be adequate to have Swissprot?
They are in Swissprot, but they don't have a G name, so their name is just the accession number.
Okay, so, and you can't search by accession number right now.
So I can access the accession number, but I cannot type it in there and get it recognized by.
Right, it's not in the autocomplete, is that right?
Yes, exactly.
Right.
So
Right.
I type it there and there is nothing.
Right.
Yeah,
Right.
exactly.
Right.
Yeah, we can look into how we can access that. I'm guessing it's just not indexing the accession number.
Right, because I don't think we have all of Uniprot available in the entity ontology to annotate.
And the other thing that I was thinking, there was a discussion about adding the interacting taxon, like in the like his input, right?
Right.
If I
That's
remember,
right.
but it's not available yet, I suppose.
If you want to add a has input, say to a process of an interacting taxon, you can do that here. Use this circle with the other actions, and you can add an extension and your input there.
But yeah, okay, it says that it has to be chemical, anatomical entity.
Yeah, let's try and
protein.
see if we can update it. So yeah, it looks like, sorry, we'll have to add that functionality in as well.
Okay.
Yeah, that's a
Okay,
more
it's all right. I thought I was doing something wrong.
Yeah, no.
Okay. Okay, cool. Thank you. But it will go there.
Yes. Yeah.
Yes. Thank you.
I had a question in relation to yesterday's talk by David. He said a nice way to begin is to put all your molecular functions in a row. But I'm finding that to get it to see it on the graph, I need to also put the gene name. I can't just put molecular functions. Is that correct? Or is there a way of just putting molecular functions next to each other without naming a gene?
If you use the add individual, you can add whatever you want. So you can add individual molecular functions.
Or individual.
Okay. I see. I was adding an annoton. Yes.
Yeah,
Okay.
so.
Got you. Thank you. Yes. Thank you.
Okay. Are there any other questions? Okay. If not, then sorry.
Harold asked if I was here. I'm here.
Okay. Okay. If there aren't any other questions, what we plan to do today then is let everybody do some hands-on modeling. We'll have everybody go on mute and work on your models here. If you have questions, see if we got the chat now enabled. Is Susie A on? Susie?
Not yet.
I'm on, sorry.
Sorry, do you know as host, can we enable the chat?
Is that not working?
It looks like it says it's disabled. So I see that you were able to post to it, but maybe us.
Let me poke around.
Okay, thanks.
It has the same settings as yesterday, so everything should be the same.
Yeah, that's really weird.
Yeah, I'm not able to post to it. So I guess as an alternative, until we sort this out, if people have questions, you can certainly add them to the agenda. I'm also on Skype. My Skype is Bannock and Kay. I'll put that in the agenda. And people can certainly, Skype questions as well.
Yeah, do we want to set up something like do people think it might be helpful to set up either another Zoom room or or something like that where we can just have, small offline conversations? Is that something we could do, is set up like another Zoom or a breakout room or something?
Maybe what would be
Anyway.
useful if we do something like that is to have some sort of quote unquote theme so that we know what type of questions we have.
Right.
Actually, now that I think about it, maybe it's just best to have, because, everybody's working at home. Like if you want to tune out, you can always just, either turn off the sound or whatever. Maybe it's best that we just use this so that people know when a question comes up. Because again, I feel like if one person has a question, you're for sure not the only one.
Yeah.
Does everyone feel comfortable with that? We can even stop recording, because I don't think there's much point in recording this whole session. And then we can just make it kind of more informal.
I think that's a good idea.
I think that's a great idea. I wonder whether, maybe the messages is working. I was going to say maybe we have to all log out.
Oh yeah, mine's working again now too.
Yeah, great. Yeah, okay. I have to go for an hour, but it's because I have another commitment. So I'll be back.
Okay. Okay, we're back to recording. Okay.
Can I delete this?
Well,
it's got nothing in now, so it's fine. It's fine. Yeah. Okay. So I searched annotations, found the one I wanted.
You got to click on.
I know what I didn't do. I was confused because it brought this here. Now I've seen.
Right.
But actually, I would have expected that not to happen.
Yeah, I actually noticed that yesterday. And so yeah,
that was what confused me, and I just hadn't seen that I needed, because this is
right.
It's just, well, I was kind of, if there wasn't a box, why is there a box there? Is that for any
So
reason?
if you have multiple pieces of evidence, you can just click.
That's sort of a select all button.
Yeah,
we probably should have a mouse over or something that says select all.
Yeah. I think because there's only one annotation appearing when I do that, I'm just assuming, oh, well, that's obviously the one I want done. But I think I just didn't see those. If there'd been a list, then it would have been more obvious that there would be. Okay, I'm sorted anyway. I'll stay on this laptop because I'll probably have more questions.
Okay.
Okay. How do I stop sharing? Okay. Kimberly.
Yep.
Sorry, I've not muted. So, sorry, I was really paying attention and I just haven't got any of this. From the table, how do I add from the table? Add, I have to create 2 activity units and then connect them in the graph, or can I connect them in the table view? I can't remember.
Right now, connect them in the graph.
Okay, so I just create another one. Okay. Yep.
I have a question whenever there's time.
That's good.
I'm not sure if Val is done or not.
I'm done, sorry.
Right, so let me share my screen. I think this is it. So you should see my Noctua screen.
Yep.
Okay, so I wanted to start from scratch, so I was going to delete my activity so that I could show you.
Okay.
So I went over to the danger zone.
Right, which, yeah. Sorry, yesterday I said, yeah, no, that's not
do it.
functional
So I'm
right
just
now.
going to start again.
Yeah.
With the same gene product. That goes at 29. And I was excited because I was like, okay, now I can, model this particular process, and I'm going to search what's existing.
Okay.
And it's a demethylase that's involved in the regulation of trichomorphogenesis. So there are these hairs.
Okay.
Leaf hairs. So I picked this one, and it's an IBA annotation. Great. Pull it in. And there was another paper that said that it was in the nucleus, and I was like, okay, I'll look for that too. And there it is. But I noticed other annotations to other complexes. I'm like, well, how do I know which one to pick? Maybe it's the nucleus, because I'm concerned about where it is in the cell, kind of at the organelle level. So that's the one I picked.
Okay.
And then I was like, okay, now process. Is there something annotated that has to do with trichomorphogenesis? And there is, but then there's also the molecular process. So now I'm like a little bit stuck. I'm like, well, which one do I want? Do I want both? Do I want all of them?
Yeah.
Does it matter? What do you pick? Like,
So
do I
this is a
pick
really
the
good
phenotype,
question.
or do I pick the thing that's been shown, kind of in a molecular assay?
Yeah. No, so this is a really good question. And we've been talking about this with the GO-CAM modeling group, right? Because often there is sort of this molecular level process, right? And then sort of bigger organismal level process. So you can nest your part ofs per biological process. So you could pick a molecular process and then say it's part of the larger organismal process. It's essentially.
Ooh, how do I do that? So let's pretend, no, let's cancel this. So it's a demethylase. So it has both methylation and demethylation. I don't know why that is, but never mind. Let's pick this one. This one's IBA. This one's mutant phenotype. Okay. Let's just pick the IMP one for now. So now I would nest it by doing add extension.
So is it methylating or demethylating?
Well, see, that's a good question. And I haven't gone back to read the papers. All I could see was that for now, there's demethylation, which is IBA, and methylation, which is IMP. But maybe we can kind of gloss over the details of this particular process right now and say, let's go with it.
Just because you'll get the demethylation for free probably because of the ontology. But anyway, but let's go ahead and model it. Yeah.
Right. So if we kind of just go with this one's right, in order to nest it, I would go to add extension and then add part of biological process.
Yep.
Is that how it works?
Yep. So now you've got a larger biological process. You can see it's nested further in. So that's a bigger process.
So let's pretend we're going to do that one. So to me, this, I'm just trying to do the visual of the nesting. So let's save it. And so and now Kimberly, sorry, but like how do I look at this in the graph?
In the graph, so click on to the left of the export as, there's a graph icon.
Oh, that's okay. Yes.
I just asked the same question in the chat a minute or two ago. It would be really, and I had a go before and I couldn't see it and I forgot. It would be nice, where it says it tells you what the view is. I'm in the graph view now. I don't know how to switch back the other way. It would be nice if just to the right of that, if you could toggle between the two really closely together, that's where I would expect the switch to be, whereas you kind of have to look around and you see this little icon.
Yeah, I agree. We also looked at the icon. We found it, and then once you found it, it's okay, but it's not.
Well, I used it a month or so ago and I forgot what it was, and it still took me about 2 minutes to find that. It should be really obvious.
Yeah, when you haven't been there in a while, you look at yeah.
Okay, we can make that more prominent.
Even in the space in the table view, there's Twitter, Facebook, and icons. I don't know why they're there. Are they there for a purpose? Because that is where I would sort of just next to.
Yep. No, I'll talk to Tremaine and we'll make the graph icon to toggle back and forth much more prominent.
I'm done.
So Tanya's question, I mean, I was sort of in that same kind of place. So as a general rule, when making the annotations, right, you know, that you want to go to the sort of more anatomical process, if you have information about that. So if you're talking about gene regulation, right, you know, that you have that gene regulation, and then in the context of the sort of whole cell or whole organism process. And it's better to so you do that in the nesting way that you just illustrated.
Yeah, that's right. That's the idea.
Yeah.
You have smaller processes nested inside larger processes. Yeah.
Yeah. How far down can you go?
As far as you want. I mean, I don't think there's a limit, is there? I think you can just keep adding. But I mean, you know, realistically, just think about the the informative,
right,
ones, and and as I said, you're already, because of the ontology, you're already going to get some of these annotations for free, right? Once you've got, in this case, the demethylase activity, or what was it? Was it demethylase? I don't know. I can't remember now, but let's just say it was demethylase activity. You're going to get to, because there's a link in the ontology between demethylase activity and the process demethylation, you're going to get all those for free, and you don't have to bother putting them in. It's a question of the other ones where it really is telling you, it's providing additional information.
And Paul, when you say you're getting them for free, does that mean in the annotation preview, I will then get that extra line in the GPad?
I don't know if we show it in the annotation preview, but we do have the function process link. So I don't know if that's in the annotation preview now.
No, it's not.
Yeah, so it's not.
Yeah,
but it will be. Yeah, there's that's in the longer term requirements.
Yep.
But I think, I don't think there's any reason to restate something that you've already stated. As long as we have the relation, the ontology, you'll get it.
So should we check that the relation exists in the ontology, or just kind of?
Yeah.
No.
See,
I think that's always good advice.
Yeah,
You're curating,
It's up to you. How much do you trust the ontology? I mean, I think
certain parts I trust a lot.
I was going to say, Tanya, I think it's pretty darn good. So, but yeah, if you have the time to check it, yeah, of course, it's always nice to have people checking the work in the ontology just to make sure that it's there.
Trust but verify.
Sorry, I've been a real numpty. I'm in the graph view now, and now I can't get back to the table view.
Okay, click on the workbenches link at the top.
Yeah.
Go to Noctua form 2.0 at the top of the list.
Yeah. Sorry, I thought I'd gone under every menu, but I obviously missed that one. Right.
Although for the future, it might be nice to have an icon in the graph view to go back
Yes.
to the form
Yeah.
view, just like
Yeah.
you have one in the form view to go to the graph
Yeah.
view.
For me, it opened in a different window, so I can just go back to the the table like just.
Yeah, no,
next
I usually have them open right as different tabs in my
Yeah, that's
browser.
right. Yeah.
The next tab, sorry. That was
Yep,
the-
no,
Yeah, it
no
launches
worries.
a different tab. Yeah, so that's probably the easiest way to toggle back and forth.
So you guys, like Kimberly and David, you guys probably have all these hacks that you have developed over time that you're just like, well, yeah, of course I have 2 tabs open. And we're like, oh yeah, that'd be a good idea.
Yeah, it should automatically open it up as a separate tab. If it
No,
does,
I mean that you
look
keep
at
them
it.
open in separate tabs as
Yeah,
part
yeah,
of your
yeah,
workflow.
Yeah. I'll put it in my notes for things.
Yeah.
To,
That seems obvious now. I see they're in different tabs and they're there, but I just hadn't clicked that would be the case.
Yeah, no, it's okay. I mean, this is exactly why we're doing this this exercise.
A comment really, and maybe it's a question, I don't know. So I try to recreate a biosynthesis pathway like David did yesterday.
Oh, great.
So I put the genes as they are acting, right? In the first step, the second step. So I'm trying to do the lipid A biosynthesis in bacteria. Just
Okay.
it came easy to me, and it is easy. So I put them in the order, and then I see in the table they have been sorted with alphabetical order, which it's okay, it doesn't matter. But when I'm going to the table, to the graph view, they have been added in the reverse order somehow. So the one that I added first, it is further to the right. If
Can you share your screen, Penelope?
Yes.
I can picture it, but.
Yeah, you'll probably just need to click and drag.
Yeah. But
Yeah,
if I enter them in an order, that it will be easier to appear.
Yeah.
Let me just share the screen. Where would that be?
Yeah, and if it's.
I think that should be. Okay. So I added everything, so I can see them here on the table. They are in alphabetical order now, but when I'm going there, it is in the, so this one should be the last step. Okay, so I was imagining it and I put it in the table last.
I see, right.
So I have to do the reverse option manually.
Okay.
It will be nice if it was like that I put them like this.
Yeah, we could try to figure out how to make that easier to do, but it's okay. I mean, you know, for now you can just connect them up and you can still connect them in reverse order. You know, you can still click and drag.
It's more like a visual thing of how we have writing. You know, how to put orders. Of course, I can.
Yeah, so we can. I don't know if we, Kimberly, is it worth showing the sort of the site escape layout tool? Is that a helpful thing for her to use at this point? I haven't tried to use it in a while, so I don't know how.
I think for linking things up at this point, not quite yet.
Not for linking, but for rearranging the diagram, like rearranging the layout.
I'm not sure, Paul. Let me look at that.
Yeah. But yeah, so we'll get back to you because we have a tool to make the layout, rearranging the layout a little easier. It's just I don't think it's been tested in a while, so
it's okay. Just
yeah, because you're right. You can click and drag these around, but it's a bit of a pain.
Yeah, and if you are doing a large pathway, I suppose it will be.
Yeah.
You know, you might be confusing something. You put it in the wrong place at some point.
Yeah. Yeah, that's right.
Okay, thank you. Stop sharing.
Okay, so we are at the top of the hour, and so on our agenda, at this point, we were going to all reconvene and go through some specific models and discussion together. I'll give everybody just a few minutes to sort of get ready to task switch here.
So Kimberly, I've got, I put a couple of activities into my model in the form, and then I went to the graph view and I could see the activities and I connected them up. So then I thought, right, I know what I'm doing. I went back to the table form to put the other activities in, and I put 4 in. But now when I go to the graph view, I can only see one of the new ones.
Sometimes when new things get added to the graph view, they're on top of one another over on the left.
Oh, right. Yeah, they are. Right. Okay. Yeah. And also in the form, go back to the form. Form is, there's a connect button in the form. Does that not work? I can't remember if you said not to use that. Does that work?
Whoops, sorry, I was on mute, Val.
Oh,
Yeah,
sorry.
so for now, right, don't use the connect button in the form.
Okay.
Do everything in the graph editor. Okay, so Petra and Bob graciously volunteered to present. Their slides are linked from the agenda, but I'm going to turn it over to you guys so you can share your screen.
Okay, I'll share my screen. And,
all right, thank you.
I'll share my bigger screen. Share. And hold on, I'll go to the slides first and I'll take it to the model from the paper. So there's a paper from 2019 and Bob will, because he brought up the paper, he will go and summarize the results for you.
Yes, briefly you can see under the stimulated tab there really what we're trying to model. This is, again, an investigation of a GTPase regulating chemotaxis in Dictyostelium. So this paper is really focused on the function of RAC-E, which again, you can see in that figure. Previously, in previous publications, the authors have shown that a RAC-E null mutant is defective in chemotaxis, and also that in a RAC-E null mutant, the phosphorylation of that AKT molecule, which is a protein kinase, that is also defective in a RAC-E null mutant. So in this paper that we're modeling, the authors found, I guess they initially found that a GDP version of RAC-E rescued both of those phenotypes. And they further found that RAC-E is phosphorylated by GSK3 in response to cyclic AMP stimulation. When that happens, RAC-E will bind, basically RAC-E will bind to another GTPase, RAS-C, and to TOR, which is part of the TOR-C complex. So RAC-E, in response to cyclic AMP stimulation, forms a supercomplex with those other two proteins, which then goes on to regulate chemotaxis. And I guess one of our questions here is, RAC-E is a GTPase, but it's not functioning as such here in this model. That is, the GDP bound version of it has to be phosphorylated for this supercomplex to form. So, you know, one of our questions is, how do we represent that in the model?
Yeah.
Is that a fair summary to get us going?
Yeah, we basically, that's why it took us a little while. And that's why when you ask for how much time, it's when you kind of, yeah, do still mostly the linear annotations and then start to model after, yeah, a couple of months not doing it or so. It always takes a little bit longer to get your bearings, basically, but it was fun to kind of try to model that. So what is the new finding is basically over here. I want to say in the beginning, harking back to yesterday, that this part here, this is what I would have liked to clone because we made it more a full model from the cyclic AMP mediated signaling from the receptor to basically these other things, a little bit like it was here. So but so what was a challenge at first was because we wanted to bring out that it's the GDP RAC-E, which is a road GTPase, but not acting as a GTPase, that this one, the RAC-E GDP, is basically phosphorylated by the GSKA kinase. And then it does bind all these other, so it does bind an activated RAS-C, which is the GDP RAS-C, and it binds TOR. And we put also the TOR complex here because it forms the super complex. I'll come back to that a little bit later. So the one question is this correctly done? When you look at that, has input GDP RAC-E, because I did that by adding, I could not do it differently somehow. I added an individual RAC-E, and then I could, up here, basically, I could add GDP, and it took it as this, and then I added the other things to make this activity model. It's the same here with the RAS-C GTP. So Kimberly, David, is this how you do this when you want to really kind of make sure that you have the specific form? Because they also showed in the paper that the GDP RAC-E is the mostly available form in the wild type. So is that the?
So I think this is great because you're actually modeling what the activity. So you know, your far right box there at the top, the signaling adapter activity. Yeah. That's the right way to think of it, which is, you know, what's its function? How is it active?
Yeah.
That's right.
You know what was basically the thing, because we didn't want the GTPase activity because it doesn't play a role in this pathway for RAC-E.
Exactly.
So we kind of looked for another function, which we of course didn't have annotated anywhere. Yeah. So and then we looked around for a fitting function, and we have actually then found, because we first thought about scaffold or so, and we found actually a term map kinase scaffold activity, and then there was no row GTPase scaffold activity or so, but a parent of that was basically signal adapter activity, so we kind of used that.
Yeah, so I think that's a great way to characterize it. And then the question is, you know, how do you say it's the GDP bound form? So you know, the way this is one of the reasons we have this arcane relation that I'm not sure is available in the tool now. This that has small molecule activator relation. Do we have that in there?
I thought it had been added, Paul, but we can.
Yeah, let's take a look. So bottom line is, you know, you want to
So
be
I
able to
added
say,
here an individual, and I can just show it.
yeah, so it's, yeah, so go ahead and add GDP as an individual right now.
Ah, okay. I did it the other way around. Okay.
Sorry, you want GDP, right? Because you're saying it's the
Well,
GDP.
yeah, I can do for RAS GDP, but I can do GDP here.
They are there, Paul. Those relations are there. They're
Okay,
not
great.
they're not at the top of the box, but if we search for them.
Okay, yeah. Okay, so if you move that over next to the signaling adapter, so basically what you're going to say is you're going to say that you know, there's an important regulatory interaction with GDP, right? That without that interaction, this isn't going to happen. So I see you've tried to capture it as an enabled by. Technically,
when you look
it's
here, do you see it as has input to the correct thing, you know? So the GDP,
that's funny.
So it shows up as a RAC E GDP and a RAC C GDP.
Oh, is there? Oh, I see. There's a RAC E GDP form in your that's.
So it shows up correctly here. That's why when I edited it as individual and paired it up and then added it basically via relation to the activity unit, it kind of worked that in the box here, it shows up kind of correctly that these are kind of together, you know? So that's why we left it like that, because it looked kind of okay, but since I've never done this,
where did you get the GDP RAC E? Is that something that you have in the? Was that?
No. What I did, I added basically the RAC E.
Ah, right.
As individual.
Yeah.
And then I went here, and I did the GDP here.
Right.
And added that. And then I had RAC E GDP.
Okay, got it. Yeah. All right. Yeah. So that's not the way we would represent a complex, but just think about so the way you probably want to do this in this context is.
I mean, this alone is not a complex. This is just the form of this molecule.
It's a particular
Right. See that? Yeah,
that's a modified form.
Right. So actually, this is a you've used the graph editor to do something that isn't legal. That's okay though. It's
really,
you know, this is why I think we're really encouraging people to use the form, because it restricts you more in the things that you can that you can do.
Yeah, but it really restricts you. So so we start with the form, but then we quickly go to the graph because you can do more things.
No, so the way we're going to try to do this is just remember what we're trying to do is, you know, again, this is an extension of Go annotation. Okay, so we want to be annotating gene products and what they do. Okay, so I can represent what RAC E does. Okay, so it's fine to say, you know, RAC E. So if I go back to that signaling adapter activity, it's fine to say it's enabled by GDP, sorry, by RAC E, but not okay to say it's enabled by RAC E. But I see how you got that. So bottom line is, I would erase all that.
Okay.
And I'd have it enabled by RAC E, right? Which is, and then you can actually say that activity is the signaling adapter activity, you know, it basically has a small molecule regulator of it, right? Which is going to be that.
Oh, so you would say.
So you can make a relationship between GDP and activity.
Regulates or positively regulates to this.
Well, no, you can't say that because so I know it's because it's not a gene product. So remember, in GO-CAM, it's just like Go annotation. Only gene products can have Go molecular functions, right? So now look for search for the term. It's not going to be easy to find, right? Kimberly just said.
I would just do your regular, you know, Control F.
Okay, yeah, do a Control F here. Yeah, sorry, this is a rare relation, but it's great that you showed. We'll make this easier in the future. This is a really rare relation, so we haven't yet made it easy to find. So if you do Control F while you're in this, you can actually do a
While I'm
find.
in this.
Yeah, go ahead and do a Control F.
F?
Yep, and you can do search and find.
Oh, it's just basically, yeah.
Yeah, just like you would on any
Command F here for me.
Yep. Okay. Yeah.
Sorry, if it's phosphorylated rack E that has the activity, can't you just?
It goes here. I cannot find anything in this box. I don't think so.
Oh yeah. No. Go ahead and do a Control F inside the box.
Command F. Apple. Yeah.
Yeah. Command F for Apple. Yep.
Yeah. But
and then look for the has small molecule regulator, or is small molecule regulator. You'll see. It's
or just small molecule.
Yeah, just search for small molecule.
Do this again because I can't do it. The Command F in this window.
Really? Oh, it works for me. Interesting.
Maybe I need to go to Firefox to do that. I'm always in Safari.
Ah,
weird.
Oh, that's interesting.
So, because that has all my things. But you say small molecules. Well,
are those alphabetical? Kind of, not even.
No, they're probably
random.
Sure, but yeah, there's a has small molecule.
Well, maybe look at the very bottom. Maybe are they like by ID?
Right. It is towards the bottom.
We even have
they're by ID.
We even have obsolete in there.
They're by ID. So let's.
Yeah, it would be great if these were alphabetical.
Yeah, sorry. This is just as we just added these, and so this is. But this is exactly.
But like there's a bit of cleanup we could do
for this case. Yeah.
Hello. I don't know if you can hear me.
Yeah, I can hear you, Antonio.
Okay, I just thought maybe I misunderstood the figure, but I thought it said that so rack E is always GDP bound, and then it's phosphorylated by GSK. So surely you couldn't you just specify phosphorylated rack E in column 16? Because it is for, I guess.
Yes,
it's
right.
So
that's why I said in wild type it's mostly as a GDP form. But
yes, it's not regulated by.
But it's still kind of new. Was a new finding and an unusual finding that this basically does not act as a GTPase there, and it's this GDP form that gets phosphorylated, and then it's a central molecule that then binds all these other things to make this super complex that activates the downstream kind of kinase.
So can't you just say that it binds whatever the other one was when it's in the phosphorylated form?
They're right there, Petra.
There you go. Is small molecule.
What is it? Small molecule.
Small molecule, yeah, has small molecule. Is small molecule
regulator.
Activator. Yeah. So
activator.
Yeah.
Activator.
But it's not regulated by the GDP.
No. Oh no. Activator. That.
Well, it will presumably a GEF will then deactivate it. So. It's probably anyway. It depends on whether you're right. Whether you think that's an important statement to make. If it's important to say it's the GDP bound form, and you expect that it could be regulated by GTP exchange, then you might want to have that in there. But otherwise, you're absolutely right, which is you want to have the target of the phosphorylation should be rack E, not GDP. And the target so the previous step, as you were saying, Antonio.
So you would
and
do
the enabler should be rack E.
So you would do the same for the rack for the racks.
Yep.
For the rack C, you would do it the same way then, that it's activated by GTP. Small molecule activator GTP. So you would always do them like this. It's the way to do it.
If you think that's important to represent in your model, but
Yeah, I think it is important because that's why they kind of made them different colors. It is that is the new thing really, because the typical thing that was found, because these molecules do different things in different settings, partially, and it's a GTPase, and then it also occurs briefly as a GTP form in other pathways, but this was basically the new part of this quite good paper, that it's the RAC-E GDP that is through phosphorylation basically activated to do all these kind of things.
It looks like phosphorylation by GSK3
yes
enables the RAS-RAC dimer to come together and to activate TOR.
That's correct.
Yes, that's correct.
I wouldn't do anything with GDP here, like specifically stated in the.
Yeah, I would tend to agree with you. It's not necessarily. Anyway, it's as I said, this is how you would model it if you think that's important for regulating the function. And I think in general, I mean, I think it's pretty well understood that these these GTPases have two states, one bound, one state when it's bound GDP and one when it's bound GTP, and each of those states has different activities. So anyway, it's sort of up to you what you want to do, but you also I think what Antonia is getting at is, as soon as you know that there's something regulating that, then you can say, oh, there's a GEF that negatively regulates that adapter activity. And so then it becomes quite clear that it must have been the GDP form.
Right.
So
we
anyway,
have this here from an older paper, because it's known that basically down from this, that there is a specific GEF-A kind of activates the RAS-C. So that's the classical kind of,
right. But again here, just think about the way you've modeled that signaling adapter. You're not activating the GTPase activity, right? I mean, if you just think about, I think the way that we've represented GTPases in Go is small GTPases is a little funny, because really they have a regulatory function. They serve basically as binding interactors that regulate something. And so so things like signaling adapter activity or molecular function regulators is really a better description of what activity it's performing. And the GTPase activity is going to be carried out when it's bound to GTP, but basically it just decays it back to its inactive state. So in other words, its GTPase activity is negatively regulating its own binding activity, right? So I think again, we just have to, I think we probably have not really thought much about how to properly represent the functions of small GTPases in Go.
I see. Yeah, I see you.
Right, because we call it a GTPase activity.
Yeah, that's why you like that we here did this, because we kind of really thought, what is it doing here?
That's right.
Right.
And if you think about that for the
as I said, this is also the new paper basically, and these other things we kind of added.
Yeah, but if you think about what RAS-C is doing, it also is really acting as a protein binding regulator.
Yeah.
Right. Yeah. That's really what its function is. It just happens to be activated by by a GEF.
Yep.
Yeah. Okay, we'll change that. Yeah, I make some notes.
Yeah, no, it makes total sense. It's just, this is just, I think you guys have picked a good example of where I think, we haven't been very,
right.
I think we haven't been exhaustive enough in how we really describe what small GTPases do when we.
Yeah, and there are open tickets about that, actually.
Yeah,
and Paul, Paul, while we're doing this, can you show us how to add that it's regulated by phosphorylation? Would we do it in the same way that we tried to do here by connecting GDP to it?
Oh, which one? Where you did the no? So you did it when you did the kinase, right? So as soon as you said
okay, so that I see. So
that's the kinase.
That's sufficient.
Yeah, so,
okay, good. All right. Okay.
Yeah. Yeah, and if you really, you know, and yeah, anyway, yeah. So that's correct. What you did is you basically said it there, because as soon as you said has input rack E, so you should make it just so that it says has input rack E and not GDP.
So should
then
you
you said that the protein kinase is acting on rack E, so that means it's the target of the kinase, so it's phosphorylating. Yeah.
Okay, good. Okay.
Okay.
You nailed that.
Inadvertently.
No, I think no, quite. I just think probably intuitively, right? Yeah.
So okay, so it would be here if we wanted to point out that it's the GDP form like this, but here we would just say that it phosphorylates rack E.
Yeah, because just think of what's the target of the kinase.
Yeah, of course, of course.
Yeah.
And Petra, one small point. I think if you want to include that GDP, you would actually have the arrow go in the other direction for the relation for this small, has small molecule activator.
Yeah, I kind of like using the reverse relation, which is small molecule activator, but we should. We really need to write documentation on that. Sorry.
Right.
I should have expected that someone would come up with the most difficult example for us. So
okay. So the rack E is the small molecule activator.
The other way.
Exactly. You say it. Yeah. So you can say is small molecule activator. That's right.
Yeah. I'll do that later then, because it's so hard to find it.
Yeah.
Okay. Okay. So, no, good to talk about that, because we found a way, but we weren't sure, you know, because it's quite an interesting paper and we liked it a lot for modeling, but and it's interesting that you say how we did it, that's more like a thing that should not be done like that. It was also because I'm just a typical trier and clicker. I try everything. I'm also a good tester of our database. I can break everything. So that's why when something goes, I do it, you know, so.
Yeah, no, but I can't blame you. I think that's fine, but that's really why we were trying to encourage people to follow the workflow of using the form and then just using the graph to connect things, because you know the graph is able to basically build pretty much any graph you want, and it's not really trying to adhere to the data model. We'll get to the point where it will adhere, but just so that everybody's aware that the tool is too flexible. And that's really why just try to use it for making connections.
Yeah, you know, it's kind of funny how, because you have been around for quite a long time, here and there toying around with it, and that's when there was only the graph, you know.
That's
so
right.
That's why we are still kind of graph oriented a bit more, but you are right, and it's kind of just getting used to do it a little bit more with the form. Yeah, because we did use the form in the beginning, but then we kind of continued on the graph.
Can I ask a quick question? I built a pathway and then I realized that I'd used the wrong relation, because when I'd done the relationship window had popped up, I hadn't realized that it was a scroll bar to the right. So I thought that the most specific relationship was positively regulates. And then I realized that hidden underneath that, there was direct positively regulates. So, I don't see that many. Why do you see all those? By default, I just saw a short list. How do you see all that?
if you keep scrolling, the list is really big. So we sort of have a
Oh, right. I didn't go down that far. Yeah, I didn't see that.
At the top, we put all the ones that we expect you to actually use, with this one exception that we.
Yeah, I just
added
didn't
these
see
new
that it was.
terms, so those aren't yet in that top section. Otherwise, you know, as I said, I think the tool is too general. Basically, it imports all of the relations ontology. So.
Right.
You can pretty much ignore that. We'll make sure that the
So what my problem was when I didn't see that there was anything below, not down in this list. I just hadn't seen. So if you go back up by default at the top view, you don't see the full list. It's chopped, and I hadn't realised. So I thought I was seeing all the relationships, and I've done everything with the wrong relationship. So now I needed to edit them, and I can't figure out how.
Are not allowed by the reasoner, so you can not.
We can do that, Val, if you're okay. Maybe we'll let Petra and Bob. Are you guys finished?
Oh, sorry, I didn't.
Yeah, let me do that later then.
And then
I'll clean that up later. So
Okay.
then I stop sharing. Hold on. But thank you for.
Maybe what we can do, if you guys are okay with it, we could, revisit this model later in the week if you've had a chance to work on things. If we can do the sort of before and after view.
Yeah,
and see what it
we
looks
can
like.
do that.
Yeah, that would be great. Thank you. Oops. So Val, if you want to share your screen, we can.
I actually think I figured it out once, but I couldn't work out how I did.
Yeah, no, there's another
couldn't repeat
maybe
it.
tip and trick here that
So-
might help save you some time.
Can you see?
Okay.
So can you see what my connection isn't very good. Can you see my screen?
Yeah.
So what I've done, I've edited these two, but it's quite a faff because I'm pulling them off and then I'm redoing them. But I know there's a way to switch them
So
over.
there's a
So
tip
I've
that
done
might help
these
you.
two,
Yeah. So
and
what
I
if
want
I
to
do
do
this? Yeah, if I do this and I need to change the relation, right?
Mhm.
So what I will do is I'll put the new relation on first, because then you can clone your evidence, right?
Right, that's what I did at one point. I hadn't realised what I'd done, but I had two lines. So got you, yeah.
And then you just delete.
And then
the
remove
one.
the old one. So
Right.
there. See what I've done? I've just
you have to
done the
scroll
list and
down.
I've gone with
Yep.
this, and then I
Yep.
haven't noticed this bit. So
Yeah.
now I know.
Right, there you go.
And then I delete the old one, which to delete them you just you just pull them off, yeah?
Just pull them off, exactly, and then it will go away. And the reason I do it that way is because
Just drive me.
sometimes I will have already put evidence on that edge, right? If I need to change it, it's better to put the new one on, and then you can clone the evidence to put that evidence on quickly. If I delete that edge, and maybe that was the only place I had that evidence will go away from the clone list too.
Right.
Yeah.
So my other question is, it's driving me crazy that I'm organizing my pathway, and then every time I come back, it's all jiggled up.
Yeah.
Can I make it? Can I stop it moving around?
So I would save it. I can't promise you that it will always come back after every action to be exactly what you have. So I think that there's definitely work that we can do to help.
Save. Where's the save?
Go to model up on your menu, up on the top, those tabs, right? Model at the very bottom. Save it.
Okay, and my other question, no, it's just moved again. I'm trying to shove it up because I know that I've got a lot of stuff to go in.
Yeah.
But what would be nice, because this is part of this is two modules going to be. This is phosphorelay, and then it's MAP kinase module here, phosphorelay module here, but all of this is going to be part of a bigger model because this style one feeds into the G2M transition, and I want to put all the different models in, but I probably won't want them in the end all in the same. So I think I'd probably do something similar to what Petra did in that previous model. So would I terminate this with oh, I know. I can just, I guess this is going to end somewhere down on CDC2, and then I can put all of the part of this I want to be G2S transition as G2S transition, and then I can do another model that ends on CDC2. And is that what you would normally do? I'm just thinking, is there a way to visualize it all together if I do it in separate models?
The G2M transition, is that what you mean? That whole process?
Yeah.
Or?
It's quite a big, because it's going to be, I don't know which is a good view. The number of modules that are going to be in might be in, I can't remember which one of these it's in. This kind of thing.
Right.
So each one of these isn't a gene, each one of these is really, that's a module, that's a module. Then this is another module. But these are modules will be quite, this is the module I've just done.
Right, right, okay.
So how to, whether I'm just trying to decide whether to try and put them all in the same thing so that I can, I want to see them all together in the end. To see them all together, do I need to put them all in the same model, or is there a way that I can sort of do this one on its own and this one on its own and this one on its own?
Yeah, for now, you know, we don't have the ability to link activities across models. We will have that, but for now, you know, you might want to just put it all on one model if you want to link it all up. You know, otherwise you can make them separate and just link them later. But you know, it depends on sort of, you know, what you want to do. But again, the models really are, you know, what's a convenient unit for you to work with from a curatorial point of view. But you can certainly give the, you know, you can give a different biological process. You know, you can have a nested biological process for the ones that are in the dotted line, for example. So you can basically mark them as, you know, with a separate sub process to say it's a module. Yeah.
That was what I was planning to do, but I just thought if I'm going to sort of organize them in this space in different areas, and then every time I come, it's spread it all over again, it's going to drive me insane.
Yeah, that's why we'll play around a little bit with there's a layout tool that makes the layout easier than doing it here. It's just, you know, like I said, we haven't tested in a while, so we can try that out.
Okay, I'll carry on putting stuff in.
And maybe we can demo it tomorrow if it looks like it's working adequately.
Okay, thank you.
Okay, that was it sort of for the more formal presentations for today. I think it's fine. We can continue on with people modeling and asking questions, or, you know, if anybody wants to present what they have so far, that would be great. Again, this is really sort of meant to be an opportunity for everybody to learn. So don't worry if your model is still sort of in progress. If you have any questions, now is a great opportunity to bring them up.
I have a general question, just because I keep on confusing myself. So that's normal. It means like I'm learning. So between molecular function, when do you use regulates versus when do you use upstream of? Is there like main rules? Like,
yeah, so Paul, you had this on your slides from yesterday. Let's see, I think you uploaded those too.
Yeah, basically, it's the way we were trying to. So again, sort of going to the Go guidelines. Regulation implies control, and so if you know the mechanism, right? So that's sort of stage one. It's so we know it's we know what the mechanism is, and if it's conceptualized as a control. Is it it's something that can control the function, the downstream function, then you want to use regulates. And if it's unknown or it's upstream of, but it might be something some pre-processing that's required for the function, and you don't conceive of it as control, then you'd use causally upstream of. So causally upstream of, is most commonly used when you don't know the mechanism. Like, this is upstream, but you're not sure exactly what the whole causal chain is.
Yeah. So if we know that this multiple step, we might not know all the specific step in between, but we know that there's some, and we know part of them. Should we use regulates because we know it regulates, but we don't have all the pieces?
Yeah, I think if you don't know all the pieces, you don't really know the mechanism.
Okay.
You don't really know if it's control. Yeah.
Okay, so you need to know all the pieces. Okay.
I think that's been our our sort of general annotation guideline for regulation. That you want to know the mechanism.
Thank you.
Yep.
I have a question about what, if any, consequences are of your things failing the Shex checks. I've got the start of a model where I have it's a sort of frizzled Wnt receptor binding activity, and it's only got a few little bits to it, but it's giving me like the red thing saying there's a Shex failure, and I'm not quite sure what, if anything, that means.
Okay. Can you share it?
Yeah.
Welcome.
Yeah. Where is it? Here we go.
Okay, so what I would do to start, honestly, is just go through it from, left to right, top to bottom systematically, and let's just take a look even without the reasoner on right now.
So I have Wnt protein binding. It's enabled by Frizzle 3, has Wnt4 as an input, and occurs in the extracellular region.
Okay.
And this is where like the red flag is coming up. The reasoner is fine. It's just this one particularly has a flag. It's causally upstream of.
So I think let's look at the relation, right? So the causally upstream of or within positive effect. So you've got that now between two activities. So I think between two activities, you'll just want to say causally upstream of.
Yeah, or actually probably regulates. Right? Because
this is Wnt and.
Right, we know the mechanism. In fact, it's positively regulates, right?
Okay, yeah, sorry, I need to look more carefully at what the second activity is.
Right, so not just the sort of weaker positive effect. Oh.
Whoops, I have to log you in. There you go.
So that I was showing someone else.
Okay.
So I stripped off my.
Yeah, so just so that positively upstream or within is for connecting to a biological process. It's basically the statement that I don't know if it's part of the biological program or it's somewhere upstream of it. So for example, if all I have is mutant phenotype information, I don't know anything about its relationship to this process, then I can use this very general term. And so that's what that relation is for. Otherwise, we know, in this case, we know that it's, these are two activities of the frizzled receptor.
Yeah.
Yeah. So
and
we know
that's got rid of that.
We know the mechanism. It's a conformational change. Yeah.
Yeah, and that's got rid of that flag, certainly.
Okay.
Yeah.
So is there a similar thing here where I have a signaling pathway upstream of a molecular function? Is that causing issues as well?
Yes, that's correct.
Yeah.
Yeah. That's right. Yeah. So again, think about, for the connections, you really want to connect up the order of activities, not processes. Because again, it's the gene ontology, and we're saying things about gene products. And so you can still say that something's part of a process. That's perfectly okay. So you're part of, I guess sorry, if you just go up a little bit in your diagram, you're part of edge to non-canonical signaling pathway is fine. Sorry, right? So that's fine, that part of edge, but the causal relationship between the process and another process doesn't follow the GO-CAM specs. I want to say, what's the next activity in this flow? So I'm saying that this Wnt receptor is upstream of RNA binding. So I want to connect activities to activities. So your box there, like I said, the part of box is fine, part of non-canonical Wnt signaling, but you can't connect that box to something further downstream because you got to connect the order of the activities. So this is a great example.
So it has to go straight from Wnt receptor activity to RNA binding.
Yep, and that's the perfect example of where I would use causally upstream of. Because even though I may know the mechanism, in this case, I'm skipping lots and lots of steps. This is the receptor, right? And so it's a great place to use causally upstream of and then give the effect direction. So I'm guessing this is causally upstream positive effect between the Wnt receptor activity and RNA binding. I am just guessing from the way you model it.
Yes, it should be.
Yeah. Right. So that's a great example of where you'd use that. It's sort of like, yeah, a bunch of stuff happens here. Overall, the effect is positive. Yeah, and then you can get, yeah, perfect. So just think of, really it's the flow of activities. What's the order in which these occur?
So is there any sort of consequence of divorcing the non-canonical Wnt signaling pathway from anything downstream? I'm just trying to think if that, I mean, if you wanted to have a chain of logic going through that, would you have to do it from process to process? Or are you saying you shouldn't be doing that at all, really? You should be doing it, yeah, you should be able to, name at least one gene product that's operating as part of each of those processes. And then you're linking up the biological processes implicitly through the linkage of the activities, right? So in other words, you can assign RNA binding to some other process, right? But the causal effects and the ordering is really between activities. Yeah, and this again just gets down to, what can we really annotate with a gene-based annotation, right? It's got to be from something that a gene product is doing, not a whole process. And ultimately, the causal link can be described at the level of activities within each process. That's our, that's our whole model of molecular biology. Yeah, I've got some more molecular functions to molecular functions.
No, that's okay, because it's not at all obvious. Again, it's it's because the graph form will let you do anything that it's tempting then to just go ahead and use its full functionality. But just think about how we the spec of the model really is to connect activities. And it and it's because we're annotating gene products. you can see, like, think about the evidence model would get incredibly complicated if we had to say, what's the evidence that this pathway is upstream of something, right? It doesn't really fit at all with the Go evidence model. So it's really, what we're really capturing is the ordering of the activities. And, and as I said, you'll get the ordering of the processes from the ordering of the activities.
So is it just the within part that makes it sort of anathema to being the molecular functions?
Correct.
So you can still be causally upstream of, you just can't be.
Right.
Correct, because within means you're part of the process. So you can't say one molecular function is part of another molecular function. I mean, unless they're carried out by the same gene product. But that's anyway. It's not the way we would express molecular function annotations in Go.
Excellent, thank you.
It's really like I said, the definition of that relation is really just it's the relationship between an activity and a process where I know this gene product is doing something, and it's and I just don't have the evidence to say whether it's part of that biological program or it's acting somewhere upstream of it, and it's able to disrupt it or to change it, but I don't know how. And so it's really a vagueness statement.
Can I ask a question?
Mm-hmm.
I've done something really wrong because mine. I'll just share my screen again because hang on. All my activity things, when I find it, cheers, that's not Noctua. Not the form, this. They've all got, I don't remember doing this, but they've all got two activities. They've all got, that looks like a parent above them.
Right, so what you've done, you've got the reasoner going, right?
Oh, I ran the reasoner.
Yeah, and so what you're seeing there and also below the annotations is inferred types. So what's happening there is, right, you didn't explicitly make an annotation to the term that's on the top, but when you ran the reasoner, it checked in the ontology to see if the statement that you had made with your, kinase activity directly positively regulates the next kinase activity, if that matches any of the logical definitions in the ontology, and it matches the definition of the protein kinase activator activity. And so when you run the reasoner, it will show you anything that's additionally inferred based on the logical definitions. And that's why you're seeing that.
A phosphoresynthase sensor kinase. See, this is what drives me mad with Go, that we have so many ways of saying the same thing, because either that should be a parent of this, or there are multiple ways of saying the same thing. Because a phosphoresynthase sensor kinase activity in the ontology will be. So I've got loads of inferences just based on whether it's a positive, whether it's it'll say probably kinase activator. I don't want to say kinase activator. I've already said that by saying it's a phosphoresynthase sensor kinase that directly positively regulates this, and then a histidine phosphotron. So here, if I put this, it's almost like this is one term, put that in my basket. And then the other term it's suggesting is protein kinase activator. And this will happen all the time. Activity. both are true, but they won't be. Get rid of that. No. I didn't get that one. So there now, we've got two things saying exactly the same thing. And that has happened for every one of the things I've put in that graph for different reasons. And it's almost like we need to get, there's two axes of classification for all of these functions, basically. Whether it's, I mean, is it, yeah, I don't know what the solution is, but I mean, I don't think we want multiple ways of describing one activity, do we?
I think ideally no, but Paul and Pascal certainly jump in on this because they've worked a lot on the molecular function refactor. I mean, I guess the question is, right, the histidine phosphotransfer kinase activity, right? If that's always an activator.
I don't know if it's in, yeah, I think it is always, that could probably, that relationship could probably be added there. But now I'd have to go through. If all sensor kinases are activators, that could be added. So if there's nothing inferred, it just puts this above us. So see, protein kinase activator, protein serine 3 kinase. I've annotated to how I could infer that from based on what I said, because I haven't said anything about residue.
But what it regulates, so if you scroll down just a bit, what you said, it directly positively regulates, right, is a MAP kinase, and that's a serine 3 kinase in the ontology.
Yeah, okay, yeah, sorry, I thought that was saying that was but then you wouldn't want to turn protein, oh, we have got protein serine 3 kinase activator activity. Oh. And we get all these out in a GAF, won't we, as well? So, well.
Yeah, no, and I'm glad you brought this up, Craig, because we've been having discussions about this in the GO-CAM, modeling working group, yeah, about these kinds of inferred annotations.
Because sometimes when it's so we kind of have rules internally where, for instance, we'll say something's a protein serine 3 kinase, but we won't necessarily say the activator residues. And here I'm not sure what this activator MCS4, I don't think we, it's because it's the kinase it's acting
Right,
on.
exactly. Yeah.
But, voila. Yeah, that, although they're not all serine 3 though, because STI1 is tyrosine, is phosphorylated on tyrosine. So some of these must also be serine 3 and tyrosine kinases. And I'm not sure whether the regulation thing makes sense to say that it's one or the other. So for instance, STI1.
But Val, sorry, it's Ruth. They looked like they weren't saying that it wasn't phosphorylating on tyrosine. It was just saying that it activates a serine 3 kinase.
Yeah, but it might be, it's so STI1 here is, STI1 is serine 3, but its regulator, which one? I'll have a look anyway. I'll have a look. Now I know what they are, but it's like, it worries me that I'm going to get all these inferences saying things that we've avoided doing because we're trying to just say the one thing rather than.
Right. So in other words, you wouldn't have co-annotated, say, to the map kinase activity as well as the protein serine threonine kinase activator activity term. Is that right? You would choose one.
Yeah, because we've done it all one way systematically.
Right.
Because if we didn't, what would happen is when you start to add different substrates,
right?
Which here there aren't so many, but Style 1's got quite a lot. If you had it both ways, when you start to put your substrates on, you would have the kinase activity, map kinase activity. This isn't a good example, but you would have an activator activity. You want to see what it's doing, and then if you had to, it would be really hard because you would have to put the substrates on two places, and that would duplicate. And you've got enough going on anyway when you want to say what the target is, what it's involved in, which stress it's doing to. You'd have to duplicate all that.
Right.
On another
Manually, right.
When you're actually only saying one thing. But I'll go through this one once I've got further and see if any of these parentage can be instantiated. But I'll probably have to ask people because I don't know. No, I think as far as we know this, NPR1, I think that only has one. I think that only positively regulates. Because it's a linear pathway, so that would be fine. We can add that one. So histidine. If Antonio, are you on histidine phosphotransfer kinase? See, what I'm worried about is this might be different in bacteria. That's what I don't know. But for eukaryotes, this could have. Oh, that one hasn't got a thing anyway. So it's phosphoryl sensor kinase, which is the one above that, I think, MAC2. That one's also, this has got two things. Right, this is one I'm trying to combine at the moment. So it's got two activities here anyway.
Right.
It's got a sensor activity and a kinase activity, but then there's a term that's combines. I was going to open a ticket about this. This term combines the kinase and the sensor functions. So I think that should probably go. And people should annotate too, but I didn't know enough about it to suggest that. Okay, now I know where they come from anyway.
Yeah.
And see. That's so. But
I think this highlights a really interesting aspect of molecular function annotations, right? And what people would like to get out of the GO-CAMs and the GPAD, and right, what we think is useful.
Yeah, so ideally, I would always want to get, for a single activity, I would want to get a single activity out, because we've got multifunctional things like this one here. I do need two activities, so I don't want different ways of saying the same thing. I only want one way of saying each thing, if that makes sense.
Can I just say something? I mean, I've come in late because I went and got some food, but the thing is, I don't have a problem personally with multiple functions. And part of that is because if you're going to be having activator activity terms, then some of the activators will be activating it because they're serine threonine kinases, and others are going to be activating it because they're tyrosine kinases. And so you can't imply, just because it's an activator, which activity it's going to have. And you can't, you know, it just seems, unless there's a definite.
I don't have an example of that. I don't have an example of that though, because.
No, I'm not saying there is one. I'm just saying philosophically, there will be activators that have different ways of activating potentially a protein. I don't know that I've got any examples of that, but I know that with the receptors, sometimes there were receptors, you know, there's different, I don't know. I haven't been doing any attention for ages, but
what I would class as different activities, but here we're talking about when things are actually the exact same activity.
No, I agree. When they're exact, if every histidine phosphotransfer kinase is is always associated with activating the MAP kinases, then I guess, you know.
Well, so the reason we've gone with the sort of kinase activator, but not, well, it depends which one it is. For these, we've tended to say go with the kinase activity. So this is a kinase, phosphorylates such a thing, and this is a kinase, phosphorylates such a thing. We haven't said this is an activator because the activator part we envisage has been captured with the relationship. So that's kind of redundant with what the relationship's telling you, isn't it?
Yes, but you can't, well, my understanding is that the Go term won't be, there won't be an activator term exported to the GPAD. So people that I work with that are doing function analysis won't get the activator aspect. They won't get grouping terms of, you know, we've got a whole load of kinase act. I don't know why you'd get that as a grouping term enriched. I've got no idea, but you know, that's the idea, isn't it? Is that you end up, you can't, for function analysis, you can't just rely on implied stuff.
Well, just because something, so the reason I thought that maybe wasn't so important was because, for instance, in the pathway I'm doing, this particular one, everything's activator, but if you look at some of the pathways, they will switch in between, and this will be activatory phosphorylation or inhibitory phosphorylation. And so the outcome isn't really dependent on whether it's an activator or an inhibitor in a signaling pathway. I think it's more important when you're talking about regulation of transcription, for instance, but here it may be, I don't know. So
I don't know either, because I know that in other ways, maybe we should just, it should just be in the process. But the other side is some proteins, you know, it's just difficult, isn't it? You want to try and create groups so that everything is annotated consistently across the groups.
So my example of where we've sort of given, let's see if I can find it. I've got so many tickets that are labelled the same thing. That when
and you, an ontologist, honestly.
It's really bad. I think it is, let's guess that it's that one. Yeah. Oh no, that's not the one that I did. Oh no, I know. Yeah, this was my mock-up when I was trying to figure it out. So here you've got sort of an activatory kinase is, what I'm trying to say is, so whether it's activating or inhibiting isn't important for the pathway outcome, if you see what I mean, because you can have, and what's really important is at the end of the day what this pathway does rather than what this individual gene product does, because it might phosphorylate this, which positively regulates some pathway, and it might phosphorylate something else and negatively regulate some pathway. So pulling out positive or negative regulators didn't seem to me as important as, also it's really hard, even when I look at this for ages, I still can't work out, I have to follow the pathway through and go at the end, oh yeah, that's, if that's, you see, you know what I mean anyway, don't you?
I do. And
Val, so it's activating the kinase activity, whether on the process level it's positive or negative, but because it's phosphorylated, it becomes a good kinase, right?
Yes, but
so in the GO-CAM, you just sort of say positively regulates.
Or activators. You just say it's an activator of the kinase, but I think that's what you meant, Colin.
Yeah, yeah. I'm not sure I'm being helpful, but I was listening in and I was thinking, you know.
I don't know if you can, because in this, for the pathway, this pathway, it's fine, but we've got some inhibitory phosphorylation as well. Some of the phosphorylation inhibits the kinase. This one,
yeah, sure.
Here's one. CDR1 phosphorylates, inhibits WEE1, CDR2. So that's where I get mixed up. The phosphorylation isn't always activatory.
Right.
Yes, which is why it's useful to maybe say these are, so if you look at this pathway, and if somebody did a differentially expressed, differential expression thing, and they pulled out a lot of the things that were inhibiting the pathway. I mean, I see what you're saying. The problem is you can have an inhibitor of something that then causes an activation of something else. And so the final outcome of the pathway is you've upregulated it, even though you've increased the amount of inhibitors.
Mm.
So, yeah, which is why, yeah, I guess why it's more important to include them in the pathway than to say whether they're activators or inhibitors.
Yeah.
But maybe we should think about that in terms of having regulator activities rather than activator and inhibitor.
Well, I think, you can say for each, but remember, for each different substrate, it can act in a different manner. So each substrate, it has, that's why I think it's important to always fill in the has input field, which is it's a different activity if it's acting on a different substrate. And so if it's the same kinase and it acts, just like, the same, anyway, the way to think of it is, distinct activities. And when it's acting on a different substrate, that's a distinct activity of the same gene product.
Which is why, to me, it makes more sense that this is the place where we're capturing what the kinase is doing rather than in here and having, we know it's an activator because we're saying here it positively regulates. We captured it here. And so this is kind of redundant. It's useful information, but then like Ruth says, you may want to get all the positive regulators out, but that to me is a software issue. Because I want it to be simple for curators.
Yeah,
that's right.
I'm inclined to agree with you, Val.
I think that's the important thing is let's think about what, let's always make this, the curation process should be as simple as possible.
We have to make this easy for ourselves because we've got a task to do. And the reason we have computers is to make the other stuff easier. So I completely see what you're getting at, Ruth, when you say your users need to do things. But at the moment, I think we have such a mess that it's not even useful. This one pathway is in such a mess. And to be honest, people don't even know all this stuff. Nobody's put this together in a model ever. So I'm still curating papers and I'm still looking. I've just found this paper. I've been looking for it for about 4 years. Don't know whether it's correct, but I'm going to have to. So I think even if we put this stuff, I don't think we can expect people to use it as it is at the moment anyway. So we'd be better just making it as easy as possible.
But isn't that the good thing about what this tool is doing, is that it's helping people to curate consistently, because not all curators will think to look for multiple activities. And so
it is, but it would be much nicer if these terms weren't available in the ontology. Force people to use these terms instead. And that would be another way to achieve consistency, because we're going to take a long time to do.
Yeah, I think we can, what as I said, I think there's a ticket for this in the ontology. But if there isn't, I think it'd be great to have, people who are interested. We can send out an announcement and have a discussion on an ontology call.
But I also suggest that, before we make massive changes and take out all these activator activities, because it's not just going to be for the kinases. If you start taking them out, you're taking them out across the board, potentially.
No, I don't.
We need
everywhere.
Well, okay.
But some pathways are good places to do it though. But
I think
they might be useful. I'll try and add them as parents if I can and they make sense, but I don't think they will in a lot of places because they switch.
But I was just going to say, if it would be nice if people actually made sure that if you take them out, does it leave things without any functions because you've said, well, because there is this problem that, one of the ways I always sell Go as opposed to Reactome is that Go is able to capture things that we don't necessarily know how everything's working, whereas Reactome needs to be putting things in the place. And if we know something definitely binds and definitely activates, but we don't actually know how it's doing that, it might just be doing it as an adapter, it might be doing it as an enzyme. I mean, hopefully we'd know it as an enzyme, but it's just, yeah.
Yeah. Well, maybe, I know I can see some of these, they could be parents, so I'll ask for those to be added. And then be the ones that we can probably work out where and if they are useful or not, where they're redundant with the other stuff we're capturing. But yeah, like you say, Ruth, things might be left without. If we haven't done this part of the modelling, then things may not be annotated.
Yeah, and if you look at if you look back far enough in the ontology history of these terms, it seemed to me that they were originally meant to cover protein binding regulators. So regulators that worked by binding to something.
Yeah, that's how I use
its activity. And then at some point we made a logical definition that was broader than that. And so I think that at least to me is how we started down this road. And then of course, then you're going to start classifying kinases, right? But the idea was originally that a kinase, because it's really modifying the protein, and that modification is what's changing its activity. That's different from something that, say, is a regulatory subunit that binds to something and regulates it while it's bound, and then when it's not bound, it stops regulating it. So I think the bottom line is I think that what's happened here is we probably just have too broad of a definition of what we mean by regulator. But I agree with you, Ruth, that we have to make sure we go back in and see if we choose to make a more restricted definition, do we lose annotations for some things that we'd otherwise want annotated? So it's definitely something to think about.
I think you're right, Paul, because I think I do use these terms definitely, but I use this for things like cyclins, protein kinase.
Exactly. Yep.
But this particular thing, MCS4, I only annotated this week, and I know I really struggled finding out what the actual activity was, which is why it ended up with protein kinase activator, and I think that came from SGD. So this is a weird one in that it isn't. Yeah, this is a kind of weird. It's not like an activator as the kinases are. It is an activator of some sort. But what we could also do is decide where there's too much specificity. Like, do we really want a term that's a protein serine threonine kinase activator, or is protein kinase activator sufficient?
Right.
I don't know.
Yep.
But anyway, I will continue and then I'll bring a load of questions.
Yeah, no, I think that was, yeah. Sorry, go ahead, Paul.
No, I think it's more important to specify use the has input to specify which gene product it acts on.
So what we do anyway here for the kinase, so we never really worry about whether it's serine threonine, because we capture that on the residue. We capture those in the modifications, and we say they're modified by. So, and to me, this is also it's too exponential to what you're really interested in, or the biologist's interested in, is which residue. And obviously you can't do that in whether it's serine or threonine is irrelevant to me. It's whether it's serine 20 that's useful. So we capture that as well and say which kinase it was. So wrapping that here doesn't, I don't know, this to me isn't very useful, but people seem to like the kinase residue part.
I think it's just that, if you read the literature, it's quite common that they will refer to a gene product as acting as a serine threonine or a tyrosine kinase. Yeah, it's commonly found in the literature. Yeah.
Okay. Yeah, that was me.
Okay, can I
Yeah,
come
thanks,
back?
Val. Yeah, Petra. Yep.
I share again because so I cleaned it up a bit so that there's not the GTP and GDP, and this has input and stuff. But this one here is the reason I doesn't like it with small molecule activator. So this is the
That
error.
probably just a reflection, Petra. I'm going to double check that. Let me see. I think that the reasoner doesn't know yet about those new small molecule relations.
Oh, I see.
And it's not even necessarily the reasoner. Like your background screen is green, so logically everything is fine. I think just the
red
behind
box.
The scenes rule engine doesn't yet know
Okay.
about those relations.
Because I tested it, I took it off and it disappears, but it is this. So this is pretty new then, this small molecule relations?
Relatively, yeah.
Yeah, it's really new.
Okay. Oh, all right, so that's just what I wanted to bring up. And because we discussed it a bit more, Bob and I, if we want to keep it there, but since, you're always a bit influenced what the paper says. And since they kind of, I think also it got into Nature because it is a quite new mechanism to use the GDP bound form of a row GTPase to have such a big role. And so that's why we wanted to have it in the annotations too. And we found it great that in GO-CAM you can kind of add the specific form of a protein. So that's why, okay, if it is, then we just leave it like that to kind of see if the red box eventually goes away. And also, Paul, I only quickly, because I didn't have much time, I wanted to listen to, I looked for another kind of function here, but it's not easy, and protein binding regulator or something so general like this doesn't exist. So we can look a little bit, more back into the paper and more into possible functions, as we found a nice one here, but I'm not sure. And since this is the GTP bound form of RAS-C, I mean, it's a GTPase. So
yeah, I think this sort of dovetails into the conversation we were just having about, what I think at least originally was meant in the ontology by I think it was protein molecular function regulator or something like that, molecular function regulator maybe, and I think it was originally meant to cover things that work exactly like this does, which is by binding. But you're right. I don't think we have clarity on a term in that like that in the ontology now. So you could use that term now. But yeah, I think this is kind of what I meant as, this is one of the, when I was giving my intro slides, one of the issues that we found as people are starting to put together these models is that sometimes we don't have an adequate representation of the functions in the molecular function ontology. So this is probably one of those
and since you mentioned binding and stuff, I also wanted to bring something up because of course in our kind of linear annotations, and this is a commonly known thing, we have protein binding annotations as this RAC-E binds RAS-C and binds TOR. So what I did here in terms of evidence, I put in the physical interaction, and then looking at the output files, the kind of current GPAD doesn't have any consequences of that. But in the kind of turtle owl, we could find the IPI annotations. So just mentioning that.
Yeah, so Petra, would you want then the protein binding annotations to come back out in the GPAD from that kind of evidence?
Well, the thing is, since we have it in our grant, when we are ready with our major overhaul and when we start working back on the Go and stuff, eventually it's still a bit down the road, but that we would like, that we will switch to GO-CAM, definitely. And so our main programmer, Sid, was always saying, OWL is the correct way to go and to represent everything. And so we will go the full way eventually. And it's still so that users, I think, will use GPAD files, for example. We don't use GAF anymore. And for that, I think it would be nice to come out. I do think it would be nice to come out in the files. If you do it like that, you know, if you put basically IPI in the right places here. Because for RAS and for Tor, because this is the major evidence. We could also put, we have also others, we haven't put the full evidences in yet, but we did that also to try it out and look at the files.
Right.
So that's what I want.
Okay, that's good.
All right.
Thanks.
That's it.
Okay, so we have another half hour on the schedule. I'll certainly stick around. If anybody has other questions, feel free to jump ahead and jump in and ask.
If no one else wants to, I'd like to show my things, but so I'll share the screen. Am I sharing my screen now?
Yes.
Yes.
So I've basically, my goal was to be able to articulate SO and Go basically, and it looks like I could, because I was allowed to make an object which is an SO object, which is sequence motif. And that's the binding site for a transcription factor, right? So I have it as a loose object here, sort of and it's called a sequence motif, and I've got an address for it, but I'm not sure how to, let's see. So for the moment, I just put, I didn't even put it here.
Can I point out, Colin, there's, you see where it says in square brackets process? It's inferring that a sequence motif is a process, which is not correct. And the reason for that is because you're using kind of part of between binding, which is a molecular activity. So this shouldn't, so first of all, this tells you something is wrong here, and I can tell it's because
that's fine. I'm quite happy to change that. So what is it then? So let me tell you, for me as a biochemist, the cis-regulatory region is a large piece of DNA that you can bind, right? The activity binds, it's binding a large region, and I'm able here to specify coordinates. I'm able to say P53 binds exactly there. So that's why I'm happy to be able to have the motif. But so if you're saying it's not a part of,
I believe in the other direction it should be, but in the other direction from which you dragged it. So the DNA binding has input.
Has input, yeah.
So this way, and then has input.
Yeah.
All right. Did you see that?
Yeah, you've got two arrows going in different directions.
I'm moving this one out. It's still not happy. So it was green, but now if I put has input, it's red.
It's probably because it positively regulates.
That's because of this guy now. All right.
We'll just keep systematically moving through, right?
But we have like so in the checks anyways. Like, is it because the so term is not recognized?
No, it has to do with a sequence motif wouldn't positively regulate transcription factor.
Oh,
activity.
okay, yeah.
So
Colin, the sequence motif is not part of this gene.
There you go.
Well, and it's not an activity. Yeah.
I know. I'm just, if I don't put my link, the sequence motif is going to disappear, and I'm going to have to.
Okay, fair enough. No, that's fine.
So I'm with you. So should I now go from here to here, or should I go from the other way, from motif to DNA binding transcription factor? And so my logic is this motif recruits that transcription factor. So to me, it positively regulates the transcription factor activity, just like the transcription factor positively regulates its co-regulator, right? It brings it to the DNA.
But Colin, we don't annotate DNA.
I want to, for Greek,
I know you do.
must, and so please let me do it. Then later you can shortcut like everybody's been doing. But right now, this is the place where I can put coordinates.
Yes, but you can annotate the protein as having DNA binding transcription factor activity, and you can then say this occurs at the sequence motif, which you then put in the sequence information.
Well, this is sequence motif.
I know, but you haven't got.
It's a materiality. It's absolutely correct.
But you're okay. It just seems like you're trying to think about it in terms of the DNA, that you want to annotate the DNA, but you actually. Well, unless somebody's changed it, but we mostly annotate proteins.
I know that. I know that. So Greek C has as an aim to be able to go from go to so and back from so to go. And there's not just the motif, but the motif is for the moment allowed in this tool, right? So I'm going with that. And we will not be able to assign transcription factors to genes if we don't say where those transcription factor binds. And they bind to enhancers. And these are specific places on the DNA. You cannot attach the DNA coordinates to the protein.
And do you intend to actually add coordinates here? So
I have.
Oh, I see. Do you want me to show how you've done that? I mean, if you.
Yeah, I put it in the comments. I put it here for the moment.
Okay, so I mean that's not really a very computable way of doing it anyway.
No.
So I'm just kind of wondering if.
But so to me, that's again a software thing. So at some point, somebody can sort of like say how to give the sequence motif an attribute, which is I find it on these 1,000 different places in the genome in that cell type bound by that transcription factor. I understand I'm going a little bit beyond what you wanted to do in this workshop, but on the other hand, this is what it's about. We need to also be able to go across ontologies.
So Colin,
we cannot avoid it.
One thing you could do, which is what we plan to do, is not to, because the sequence, so if I've got this right, so what we've been doing is on our RNA polymerase sequence specific binding terms, we've been adding the so term, which describes the motif, which is part of what you want, and you can do that anyway. That's perfectly legal, isn't it? But what you're missing is the coordinates.
Well, I have them. I've got the coordinates.
What you're missing is the coordinates attached to this annotation, but really you want the coordinates of every known version of that motif.
No. I have files where I can say in that cell type.
You want it for the specific gene that you're looking at in this particular.
Let's, you can't oversimplify the whole thing, right? So some enhancers are extremely far away from one gene. They're actually controlling 5 different genes in that same topologically associating domain, right? They're so-called locus control regions. And so I want to be able to sort of say that locus control region is here. All of these genes are in the same regulatory unit, which I call a TAD. And so potentially all of these genes are subject to this locus control region.
Okay, I
so it needs to be a thing that, or at least it goes much further than that. So of course now here, I guess, or somewhere here, I could sort of like put my coordinates, right? I could sort of like put chromosome 6, normally the thing will be, so here we have the genome version. Then,
I would still put it as a comment.
And from where to where on that chromosome.
Yeah. So Colin, I'd still put it as a comment right now because, yeah, what you filled in is for relations. So yeah, I think.
So you agree with my thing here that I could put it here on the comment for the moment?
I think for now you could put it on a comment. Do that.
Not on the
relation. So yeah, close that box.
It
and
was
then,
here.
Okay, let's do that.
Yeah, here I have the
down. Yeah.
So this is where it would go.
And if I'm understanding Chris correctly, I think maybe what he's getting at is that in the future we may be able to help people do this in a more systematic way, but
yeah. I mean, I would, if I was doing this, I would put it on the actual sequence motif instance itself.
That would be fine. Just now I can't. I don't have a place where I can put it in and let it.
Oh, you don't even have the ability to add a comment here? Okay.
No.
Yeah.
Let me.
But yeah, I mean, there is this tension between, you know, doing things in a very kind of standardized way that we do with Go annotation, and the overall framework allows you to be more flexible here and do things like this. So
so you would rather have me draw an arrow from here to here than to sort of like make the to go from. So for the moment, I have sequence motif is a material entity, and it somehow is linked to this function, right? This function cannot happen without a motif.
Yeah, so but I think you've modeled it correctly in GO-CAM, which is the function belongs to the gene product, and it's acting on a particular stretch of DNA. So that's correct to use the has input is basically our way of specifying the target that's acted on. So that's correct.
And now I would like to link this to P53 or to the, in fact, I just put the activities here, right? And so above I have proteins. So I would like to be able to link this to the activity. And so the question is, what kind of relation should we put here?
Yeah. So again, I think that it's also okay to specify, since you said it's a DNA binding transcription factor, you can actually specify one of the inputs is that same sequence motif. So in other words, you don't have to say that it's the DNA. In other words, you've already said in the overall term that it's DNA binding. So I think it's okay to just put in here that has input.
Yeah, like
right?
This.
Yep. And then separate, and then I would separately say, the gene that it's acting on.
Yeah, that's here, and so that I want to.
And you can use that as a separate has input.
Right? You would actually link this one also this way.
No. I'd say actually that the DNA binding transcription factor activity from another viewpoint can be thought of as acting upon the CDKN1A gene.
Well, that's my next step, Paul. So now I want to go a little bit more granular and to say it's not just the transcription factor. The transcription factor activates the co-regulator, which then, if we just go into functions, then it activates this function, but it's via this.
Gotcha. Okay, so what's the transcription factor activating? Sorry, which transcription factor is acting?
This one, P53.
Okay, so so that's going to be the enabler. So you're going to drag from DNA binding transcription factor activity. But again, if you do this in the form, you don't have to think about the way it's connected.
Yeah, but I guess.
Yeah,
this
that's okay.
Allows me to understand it. And so this is again a positively regulated.
Enable, and this is enabler, right? This is the thing that's doing it. So go scroll up to enabled by. Yep. So you're saying this activity is enabled by this gene product, right? So that's the one doing the activity. Yep.
Oh, he's in there now. I see. Yeah.
Yep. Now it folds it in there.
And so the same will happen here. So that's also the enabler.
And so is it? Yep. And then that's your transcription co-regulator. So drag from the co-regulator. Now the other way around.
Oh, I see. Yeah.
Sorry, no, the other way around. Drag from the regulator. Yeah, so it won't let you do that because. Yeah,
I got it. Yeah. Or I should have gotten it the first time around. It's just
that's okay.
Reverse thinking. And so this one here again then enabled by, right?
Right. Correct. Yep.
So I think this to me is a huge advance. I mean, this is something we really have been wanting to be able to do for so long. The last thing would be that somehow, I would like to be able to park my comments somewhere else.
Right, like I think we in a previous version of the model that we probably did in one of the Greek C workshops, I think we had has input ensemble. I had kind of made some.
Yeah, I think it was here. Where did you put it?
Well, I had like has input, like I hacked it, you know, I just, because this tool doesn't do a lot of checks.
Yeah.
And so I had done like has input sequence, like, so let's say the sequence motif and has input, you know, ensemble, and then like some bogus genomic coordinates. But I think what you want to see is like chromosome 2 position X
Yeah.
to Y. Yeah. And so I think this can all go in the box of the binding, like to describe its input.
Or I would, if we are going this way, I would say this is a property, the coordinates are property of the sequence motif. And how I would do this, and this is going away from how we would do it in Go, where we don't have this level of granularity, but how we do it here is I would use an existing vocabulary like Faldo for, so you've got separate properties for start, end, reference, you know, strand. And then, you know, if someone wanted to, and I don't think we've got resources for this in Go, but if someone wanted to, they could easily extend the graph editor such that you could have such that when it's some kind of so entity, you could enter in those coordinates in a normalized fashion. I might still have, we still not have a causal link between the DNA binding or some kind of like way of connecting these, not just via the motif, Kimberly?
You mean like a has part or part of between? Yeah.
That's what Pascal did. Pascal did DNA binding transcription activator as part RNA polymerase. And so I found that very funny because if I do this here, but maybe now it will work. But earlier on when I was doing it, I was getting again a red. And
that was probably because of the relations.
Because of the other things I had in there. Right? As part. And then space, yes, here, this one, right. Which is a very strange one. It's about occurrence, this one, right? But now I can do it, yeah. So this is when you don't know the place, but you know that transcription factor activates probably via this function of binding DNA, a gene, but you don't know the motif, then you would be using this route here. But if we come from sequence files, from next generation sequencing of chromatin, IP, and all kinds of other assays, right? Then this is really the input. This is going to be the data that we want to feed to the model. It's not so much that it's a binding factor or that it's binding these regulatory regions. We know all of that, but the variable or the stuff that is, you know, that has to be digested is all the different coordinates. And there's sequence motif instances or binding event instances. But yes, to me, I've done my homework now. So thanks a lot for the feedback.
Good. Thanks for presenting, Colin. I think that it's nice to see what you want to do, right? What you want to be.
Yes, and it's reasonable apparently. So I guess I can now save it.
Okay. What's that? What's this valid invalid thing at the bottom?
I think that's coming from the red box, and the red box is coming because right now in the SHEX, a so term isn't valid input.
Got it. Yeah.
Yeah. So these two red ones here are just due to the fact that I put a so term in here. But yeah, no, this is great, and it's one of the few ones because I also tried to put the SO for cis regulatory module, but that one didn't work. Let me see. Oh, I'm sorry, I have to get my other window working for me. So I had also from SO the cis regulatory module. Let's see. So that's SO 727, and that one. If I try to add that one in, it doesn't accept it. See, this doesn't work. Whilst I could do the 1637 sequence motif. So somehow I'm very, very glad that right now Noctua accepts sequence motif. It would be nice if we had a few more of the Greek C SO terms accepted in Noctua.
Okay.
So David Sand, who was on the call yesterday, but today he couldn't be here.
Right.
We have now a short list of, I think it's less than 20 SO terms that we would like to be able to use for in the context of Noctua.
Okay.
Yeah. Right. So stop sharing now. Thanks a lot. Thanks a lot. Thanks,
Colin. Yep.
Yep.
sorry if I missed this, but when you run the reasoner and everything turns red,
Mm-hmm.
how do you see where the problem is?
Yeah, okay. So, right, that's something we're hoping to put more user-friendly reports in. Probably the best thing to do is like what I did with Malcolm, and is just really start systematically working through the model. If you don't mind sharing your screen, we can do that together.
It's quite a simple thing. I didn't do a big. Hang on. Share screen. I can never find that in other.
Okay.
So.
All right. So let's.
This is the shikimate pathway. So you have these two enzymes.
Okay.
And they're allosterically regulated. And they're the same thing, just two genes. And they activate. Well, they provide input for this thing, which provides input for this thing, and so on.
Okay, so over there on the left, let's just look at the tyrosine, for example. How does that relate to the function in your model?
I just put directly negatively.
Okay, so that's probably the issue. So remember, as Paul's explaining, what we want for the GO-CAMs is an activity flow. So one activity might regulate the next activity. So in this case, what you're saying is there's a tyrosine entity essentially that's regulating an activity, right? And so,
oh, right. Yeah.
You want tyrosine sensor, Antonia.
Yeah, so you can use the small molecule, regulator relation, or you can, use a sensor activity and then have that activity regulating. Yeah, either one's okay.
So I changed that, right? So I either make a molecular function with a tyrosine sensor. I don't think we have that term yet. Or I've just put another relationship in.
Right.
Okay. I'll give that a go. It's gonna take me a while to figure out how. So
no pressure.
Sharing is
your own time.
I cannot blame you.
Okay, so we've got about 10 more minutes left. I'm happy to stick around again if anybody has any questions. But for tomorrow, I think essentially we'll follow roughly the same kind of format where we can do a little quick review of things in the morning, and then people can go off and do their models. We will be around for question and answer. One suggestion we had is that we actually don't stop recording, because sometimes what we're discussing in these sessions is actually really interesting and important. So I think we'll keep the recording running during that, even though there may be some quiet time. And then I'm going to double check with Sabrina to see if she would be willing to present her slides and her model tomorrow, because we talked about that quite a bit. If anyone else would like to present some more, that would be great. I think we're getting a lot out of looking at the various models and that people are presenting. So once again, thank you very much, everybody, for attending. The feedback and the interaction has been really, really helpful and good. And I hope everybody else is getting something out of this too. Paul, is there an Alliance Pathways call today?
Yeah, there is.
There is? I thought all the calls were supposed to be canceled. I was just going to cancel the Pathways to Go one.
Oh, were they?
We said we said Go
Alliance.
calls, but we
Yeah.
didn't specify about the Alliance calls.
Yeah, I think we were going to run the same suggestion by the Alliance PIs this
Oh,
week.
nice. I think that will be well received.
Yeah, seriously, it's a lot of meetings.
Okay, I'm going to sign off and just take a couple minutes before the Alliance Pathways call. So thanks, everybody, and talk to you tomorrow.
Thanks a lot, everybody. Great questions.
Hi, everybody.
Yeah, thanks. Good end of the day.
Bye.
Thank you.